And
Yeah, absolutely.
trying to jump up the walls.
Yep.
Okay, I think we're ready to get started. If you're ready, Tricia, I'm just going to start off reading the land acknowledgement. So we acknowledge that the lands on which Memorial University's campuses are situated are in the traditional territories of diverse Indigenous groups, and we acknowledge with respect the histories and cultures of the Beothuk, Mi'kmaq, the Innu, and the Inuit of this province. We encourage everyone to reflect on these lands from where you are located and the Indigenous peoples for whom these lands are traditional territory. So with that, I would like to welcome Dr. Patricia Thibodeau to give us our seminar this week. I'm really excited. Tricia and I have worked together before, so I'm really excited to listen to her talk. So now it's your turn, Tricia.
Thank you, Colleen. All right, I will go ahead and share my screen. All right, can you see that?
Yes.
Great, thank you. All right, go ahead and begin. Well, hi, everyone. Thank you all for having me here. As Colleen said, my name is Patricia Thibodeau, and I'm currently a postdoc at the University of Rhode Island. I wanted to acknowledge the lands that I'm currently presenting on, which are the lands of the Narragansett people. And today I will be riding a wave of variability through plankton time series from the Southern Ocean up to the Narragansett Bay in Rhode Island, where I am right now. And I'll be talking with you about some of the research I did during my PhD at the Virginia Institute of Marine Science, as well as what I've been currently doing for my postdoc at URI. And throughout my scientific career, I've had the opportunity to analyze extensive time series data sets, and ecological time series are particularly valuable when we focus on plankton dynamics, because their short generation times and limited mobility make them great indicators of both short-term and long-term change. Computer's loading, sorry. So one aspect of change that time series record is temperature, and most of you know that the Earth is warming, but I really like this illustration that shows it visually. We have the long-term global average temperature up here on the right, and then the temperature anomalies for each country around the world since the early 1900s. And any bar below the zero line represents below average temperature for that year, and any bar above the zero line represents above average temperature for that year. And you can see that right around now in the late 90s, the temperature bars are getting increasingly redder and bigger. And the last year in 2016, we can see that the United States up here on the left has one of the biggest and reddest bars in the world. And one ecological response to this long-term warming are shifts in phenology. And phenology is the study of annually occurring life history events. And the New York Times published a really nice illustration of phenology, looking at the timing of spring foliage in 2017 in the United States. And so spring began to bloom in January in the South, and then move forward beginning in as late as May in the Northeast and Pacific Northwest. And when we compare the timing of that spring foliage in 2017 to an average timing of spring foliage over 30 years, we see that there have been significant changes in that in the far south, the spring foliage is occurring up to 10 days later than it used to 30 years ago, as opposed to the Midwest and the California Pacific Northwest. Spring foliage is now occurring up to 10 days earlier than it used to 30 years ago. And This brings me to the time series work that I've been able to do, which enables me to both look at long-term changes in phenology as well as other shifts due to long-term warming. And the Narragansett Bay is one region I'll be focusing on today, which is the largest estuary in New England, and it's been experiencing long-term warming. This figure shows annual water temperature on the y-axis over time, and it's seen a mean water temperature increase of 1.4 to 1.6 degrees centigrade over the last 50 years. A very different region of the world along the western Antarctic Peninsula is also experiencing long-term warming. To orient you, the Antarctic Peninsula is located across the Drake Passage in the country of Chile, and it's been experiencing long-term warming at a rate of about 1.1 to degrees centigrade per decade. And that represents an overall temperature increase of 7 degrees centigrade since the 1950s. And that's 5 times that of the global average. And this figure here is showing winter temperature on the y-axis and year on the x-axis. And we can see a long-term increase in that winter temperature over time. This region is also experiencing long-term changes in sea ice. This figure has sea ice duration in days over time, and we can see that the sea ice duration has declined over time due to later sea ice advance and earlier sea ice retreat, such that we now see an overall decline of 3 months in sea ice season since 1979. And and what's great about, or fortuitous about these locations is that while they are experiencing long-term warming, there are time series located at both of these regions to monitor how these long-term changes in temperature and sea ice along the peninsula are affecting the ecosystem there. So in Narragansett Bay, we have a long-term plankton time series, and along the peninsula, we have the Palmer Antarctica Long-Term Ecological Research Program. So knowing about these long-term trends in warming and the time series data sets available to me, I've been able to ask the following questions. First, what are the effects of climate change on pteropod ecology and physiology along the western Antarctic Peninsula? And pteropods are these beautiful ocean snails that I will talk about more in a minute. And secondly, what are the long-term changes of phytoplankton abundance and bloom phenology in Narragansett Bay? And moving to the first question, looking at pteropods, as I mentioned, they're open ocean snails, and pteropod in Greek means winged foot. And so they're commonly called sea butterflies. And this video may give you a better sense as to why. I hope it's coming through clearly on your end. These are of the species Limacina helicina antarctica, which is the third most abundant zooplankton along the Antarctic Peninsula. And the food they eat actually makes them appear very black in color. And so when I show this video, people often tell me they look like bats more than butterflies. But I also creatively think they look like the snitch in Harry Potter. But they're also a very ecologically relevant organism. As I mentioned, they're very abundant along the Antarctic Peninsula, and they've commonly been referred to as bioindicators of ocean acidification because their shells are composed of calcium carbonate in the form of aragonite that's considered a more soluble form of calcium carbonate. So their shells can be used as indicators of more acidic conditions in seawater. Along the peninsula, they're also very important grazers. They have some of the highest grazing rates that surpass that of the Antarctic krill. To feed, they use a mucus web, and this is one of the few images we have of a pteropod feeding in the open ocean. This is of a different species called Coralla, but you can see the mucus exuding from its mouth and the different particles that it's catching on the mucus, and then it retracts that mucus and eats the particles through its radula. And it's also an important prey for higher trophic animals along the Antarctic Peninsula, especially seabirds and fish. But when I was beginning my PhD, I realized we still didn't really have a very good understanding of their general ecology and physiology, especially in the Southern Ocean. And so this led to my overall research goal of determining the environmental controls on pteropod ecology and physiology along the Western Antarctic Peninsula. And to to answer this research or achieve this research goal, I had three primary research questions. Are there long-term changes in pteropod abundance and distribution related to environmental controls? And this work was recently published in Limnology and Oceanography. Second, have Limacina phenology patterns shifted over time, and what environmental conditions control these patterns? And this work was published in Deep Sea Research last year. And finally, how do changes in environmental conditions along the peninsula affect Limacina metabolism? And this work was published in GenB last year. And so focusing on this first question of long-term changes in pteropod abundance and distribution, I utilized an extensive time series data set that's been collected as part of the Palmer LTER. And every year since 1993, this program has embarked on this beautiful orange ship called the Lawrence M. Gould. And we go down the Antarctic Peninsula and sample a series of stations shown here in white in about 24 to 30 days. And we're really interested in capturing this north to south climate gradient that's developed along the peninsula, where the northern part of the peninsula is now considered a subpolar climate that's warmer and wetter than it used to be and no longer has year-round sea ice. The south region is considered our transition zone, and then the far south region is considered our truly polar climate. It's still cold and very dry. We're also interested in capturing this coast shelf slope gradient, where our coast is considered a highly productive region, the shelf is our transition zone, and then the slope has relatively low primary production and has more influences from the Antarctic circumpolar current. I also wanted to highlight this moored sediment trap shown here that's located off the northern shelf of the peninsula, and I'll be discussing this further in the second question of this talk. In terms of zooplankton collection, we use a 2-meter by 2-meter frame net that's deployed off the back of the ship at each station. And then we collect the zooplankton in this cod-in shown here. And depending on what we're trying to sample for, especially when I wanted to try to get pteropods for my experiments, we would occasionally do dances to appease the zooplankton gods so that they would fill our nets with abundant pteropods. It only works sometimes. In terms of the results, what we saw overall was a long-term variability in pteropod abundance, but no long-term directional trend. So this figure here shows the Limacina abundance anomaly for each year. So any bar below the 0 line represents below average abundance for that year, and any bar above the 0 line represents above average abundance for that year. And we see an oscillating trend over time, but no long-term directional trend. And we found that climate oscillations had an important effect in explaining this variability. And so this figure here shows the multivariate ENSO index, or MEI, which looks at the oscillating pattern between La Nina and El Nino, and then Limacina abundance anomaly on the y-axis. And we see that when the MEI is more positive or favors an El Nino year, Limacina abundances are typically lower. And so conversely, we see that La Nina years support higher pteropod abundance. And we believe this is associated with sea ice because La Nina conditions typically lead to shifts in winds that cause lower sea ice conditions overall. And so we believe that those lower sea ice conditions are actually enabling pteropods to feed more and therefore support their abundances. We also saw that temperature was an important factor. This figure has sea surface temperature anomaly on the x-axis and Limacina abundance anomaly on the y-axis. And we see that with higher sea surface temperature, there were higher abundances of pteropods. And so overall, we think that ice-free warmer waters best support higher pteropod abundances. And so despite this long-term warming and sea ice loss we've seen along the peninsula, Limacina is actually doing okay, relatively speaking, during this last 25 years, which is a good thing. Moving on to the next question, have Limacina phenology patterns shifted over time, and what environmental conditions control these patterns? And to answer this question, I was utilizing a sediment trap, which is this giant cone shown here, and I'm standing next to it for scale, pretend I'm not in my slippers. And this sediment trap is deployed off the northern shelf of the peninsula, and it's bottom moored and rests about 170 meters below the surface. And what's really great about this trap is that it's deployed year round. So unlike our annual research cruises that only happen in January of each year, this sediment trap allows us to get year-round sampling of particulate organic carbon flux and pteropod shells. And one of the reasons that we collect pteropods in the sediment trap is because their shells make them negatively buoyant. And we think what happens is that as a defense mechanism, they'll commonly retract into their shells when they think there's a predator nearby. And so we think that they hit the sediment trap and go, oh no, a predator, and they sink into their shells and then they sink into the trap. So we have a really nice time series now of 14 years of pteropod shells. And the way we collect them is with a series of cups that rotate throughout the year at timed intervals, like a carousel. And we've been deploying this trap since 1993, but for the purpose of this study, I was only analyzing cups from 2004 to 2018. And getting a little more into the methods, we picked, counted, and photographed shells from the sediment trap samples. These are some of the images of the shells from the samples shown here. And then we would measure their diameter across the shell. And this is a photo of your very own Colleen McBride, who helped me for 2 years while she was an undergraduate at William & Mary. And I realized that Colleen counted and measured over 10,000 pteropod shells to help make this project possible. So I am forever indebted to Colleen. Thank you so much for your help. Some of the results from all of our hard work. This is an image of the size frequency histograms for shells from, as an example, this is just for 1 year, 2017 to 2018. We have length here on the x-axis and then the counts or the frequency of the lengths on the y-axis. And as an example, for 1 year, what we typically found was that pteropods would begin to show up in the sediment trap around May or June. They were very small in size. And we'd see that they would grow throughout the winter season. And this was really exciting to see because there was no documented evidence that pteropods actually grew during the winter time. And then we would see that they would continue to grow into summer and then typically not be collected in the sediment trap in January or during the height of the summer because we were in the southern hemisphere. And what we believe is happening is that at this time, pteropods are feeding in the surface waters, and so they're not migrating deep enough to the sediment trap during this time because all the food's at the surface. We would occasionally catch some larger adults that were 6 to 7 millimeters in size. And so what this indicated to us was that these adults were feeding and then spawning in the late summer and then ontogenetically migrating into deeper waters, which is a seasonal migration that some zooplankton undergo, especially in the poles. And their larvae would then be collected in the sediment trap around May or June and then grow into adults in the next summer season. And so one question I was interested in answering was whether this time of appearance that the pteropods showed up in the sediment trap each year was changing over time. And so this figure here shows the day of year that the pteropods first begin to show up in the sediment trap from 2004 to 2018. And we see a lot of variability in the time of appearance, such that in 1 year, such as in 2006, they showed up really late as 250 days into the year. And then the following year, they showed up really early at 50 days into the year. And so overall, we saw a lot of variability, but no long-term directional change in their phenology or their time of appearance, indicating that overall, despite long-term changes in warming and sea ice loss, the phenology of pteropods has remained relatively stable. We found that sea ice was an important predictor in explaining this variability. This figure here shows sea ice retreat anomaly on the x-axis and the time of appearance on the y-axis. And we found that earlier sea ice retreat typically corresponded to earlier appearance. And we believe that's happening because with earlier retreat, that means that there's more open water for the pteropods to feed. And so then they're subsequently growing and spawning earlier in the year. And so then we begin to see pteropods appearing earlier in the sediment trap. Finally, focusing more small scale at Limacina metabolism, I wanted to understand how these long-term environmental changes might be affecting their physiology. And so one of the effects of this long-term warming and sea ice loss that we've seen along the peninsula is a shift in food availability, or chlorophyll A. This figure here shows the change in satellite-derived chlorophyll A comparing the early 2000s to the mid-1980s. And what we see is that the northern part of the peninsula has seen a decline in chlorophyll A, whereas the southern part of the peninsula has seen an increase in chlorophyll A. And the reason is because in the northern part of the peninsula, there's no longer year-round sea ice. And this loss in sea ice has led to an increase in mixing in the region, as once ice-covered regions are now exposed to wind. And subsequently, the phytoplankton in this area are mixed below their critical depth, so they're not able to photosynthesize and bloom and produce. In contrast, there is still sea ice in the southern part of the peninsula, but however, when it melts in the summertime, it releases phytoplankton from light limitation, and coupled with increasing temperatures, we've seen an increase in phytoplankton concentrations in this region. Thus, I wanted to look at the effects of shifting temperature and food concentrations on pteropod metabolism. And so to do this, I set up a multifactorial experiment where I did mini pteropod dances to elicit enough animals to do these experiments. And then I would collect at the same stations with our CTD water that had in situ chlorophyll concentrations, which I considered my high food treatment. And then I would dilute that chlorophyll concentration down to a 10% concentration, which I would refer to as my low food treatment. I would then acclimate the pteropods for 2 hours in their respective high and low food treatments, as well as their low temperature condition or in situ temperature condition at 1 degree Celsius, and then anticipated high temperature condition of 4 degrees Celsius. And then I would incubate the pteropods in these conditions for 12 hours and conduct metabolism measurements during this time. In terms of measuring pteropod metabolism, I was measuring their respiration or oxygen uptake, as well as their excretion. And so to measure their respiration, I would use this fiber optic cable shown here, and it would shine a red light, just like the laser pointer I'm using, at the oxygen molecules and be able to measure the concentration of oxygen in the bottle as the pteropods were swimming. And I would measure that every hour. And then at the end of the experiment, I would filter out the chlorophyll and the pheopigments from the water and collect the filtrate so that back at bins I can measure dissolved organic nitrogen, dissolved organic carbon, ammonium, and phosphate. Briefly, to go over some of my results, overall I found that increasing temperatures led to an increased metabolic rate, but there was no significant effect of food on pteropod metabolism. This figure here has temperature on the x-axis, respiration rate on the y-axis, and then the corresponding food condition in the color bar here. And we see that with increasing temperature, there was an increase in respiration rate, but no significant effect of food. And I saw the same result for most of the excretion rates I measured as well. And so overall, we can see that at least on the short term, temperature is an important factor in pteropod metabolism, but not as much food. And so one of the overall conclusions of my dissertation is that I argue pteropods are not only bioindicators of ocean acidification, as we've originally argued and expected, but they can be bioindicators of climate change in general, because we see that they're responsive to changes in sea ice, particularly in relation to their phenology, as well as increases in temperature, particularly related to their long-term abundances and their physiology. And all these factors are superimposed over subdecadal oscillations, which have an important effect when we think about pteropod long-term abundances, particularly in relation to El Nino and La Nina. And so the fact that pteropods are indicative of so many different environmental factors makes them a really awesome group to study. And this is why I think pteropods are so terrific. So shifting gears now, we're going to move up to the Northern Hemisphere and to Narragansett Bay, where I am now at the University of Rhode Island. And as I mentioned before, this region is also experiencing long-term increases in water temperature, as well as declines in nutrients and increases in water clarity. And these changes have affected the food web in that we now see an expansion of warm water fish species and increases in the residence periods in Narragansett Bay. This is seen particularly for warm water fishes like the butterfish shown here. We've also seen changes in the zooplankton communities. We've seen recently that warmer winter and spring temperatures lead to higher abundance and duration of jellyfish, particularly Memiopsis ladii, shown here. And there's been indication of a long-term decline in chlorophyll. This was recorded by Nixon and colleagues in 2009 for summer, in which they utilized a data set that covered from 1970s to 2006. However, we actually have a 60-year time series of chlorophyll A that hadn't yet been fully harmonized and analyzed. And so that was one of the primary goals of my postdoc when I first got here. And these data are all collected through the long-term plankton time series in Narragansett Bay. And to orient you to this location, the time series is located in the lower west passage of Narragansett Bay. And the Graduate School of Oceanography, where I work, is located just south of there near the mouth of Narragansett Bay. And since 1959, staff and students have gone out weekly to collect a series of measurements, which include phytoplankton counts and biomass, as well as some for zooplankton, nutrients such as ammonium, phosphate, nitrate, nitrite, and silicate, and a suite of weather data and temperature, salinity, and water clarity. And this time series was first started by Ted Sameda in 1959. And now we have graduate students that go out every week and collect these measurements. And I had the pleasure of getting to analyze all of the data that they've collected. And one of the first goals of my postdoc when I began in January 2020 was to harmonize 60 years of data and make sure that the methods were consistent. I also had to literally go back into a closet to try to find some of the original data that hadn't been digitized yet. But once I had all of the data available and QA/QC'd, I was able to ask a series of questions. First, what are the long-term trends in nutrients in chlorophyll A? Second, has the timing and duration or the phenology in phytoplankton blooms changed over time? And finally, what environmental factors are related to bloom phenology trends? And as some of you may know from working with time series, there can often be issues of missing data. And so this was true for the plankton time series that I was working with. And so in order to handle this, I utilized a Bayesian class of models called dynamic linear models. And I collaborated with statisticians at the University of Rhode Island to do this. And what's great about this series of models is that you can interpolate the missing data with uncertainty, and it utilizes the existing structure of the data you have to interpolate those missing values. And so as an example, we have the raw or observed data for ammonium recorded from the time series from 2004 to 2019, and the subsequently modeled data based on the structure of the raw data originally. And then you can separate out those model data into long-term and seasonal components shown here. And what's great about this is that you can then begin to extract some of the long-term trends that might be related to climate change and whatnot. And This series of models is also useful because any parameter and component can vary with time, and this is particularly important when we're trying to answer questions about phenology. And for the purpose of my talk today, I'll be using a mix of both observed and model data, and I'll let you know when I'm going back and forth between the two. Once I had, and so moving on to my first question, what are the long-term trends in nutrients and chlorophyll A? I first answered this question by looking at average annual concentrations of nutrients throughout the time series. I know these data are observed values and not modeled because I wanted to get a better sense of the magnitude of variability in this data set over time. And these data are shown as annual average concentrations and one standard deviation above and below the mean. And we see that for phosphate, they've significantly declined over time with a mean concentration of around 1.5 in the 1960s down to less than 1 today. We also have seen a long-term decline in ammonium concentrations. This time series only began in 1972, and we see that there is more variability in this data set, but there's still a significant decline in this molecule over time, or this nutrient. And for nitrate, we see even more variability in the data. We see that the 1980s and mid-1990s were particularly variable compared to the late 1980s and the last decade. And we see that silicate has significantly increased over time from about 10 micromolar to about 15 micromolar. And overall, these data agree with what's been observed in the Bay and in other regions that have been sampled. And some of the reasons that we see these long-term declines is because of changes in nutrient waste management that have occurred, particularly in the upper regions of Narragansett Bay. We also see that, I don't know why this X is there, but okay. We also see long-term declines in chlorophyll A. This figure here shows chlorophyll A. Colleen, is there a reason that there's people writing on the screen?
No, actually, if I don't know and I don't know how to remove it, but if you're writing on the screen, then I'm gonna have to remove you. I'm not sure. We haven't had this happen before.
Okay, just adds a little color to the slides. So I'll continue on. So we saw a long-term decline of chlorophyll A. This figure here shows average chlorophyll concentration for each year and one standard deviation above and below the mean. And we see that the chlorophyll has been significantly declining over time. And we also see that chlorophyll intensity, or the maximum value of chlorophyll A each year, has also significantly declined. And this is important when we start to think about phytoplankton blooms and the fact that we're not only seeing the magnitude of chlorophyll A decline, but also the intensity. I then wanted to see how the seasonal, whether declines in chlorophyll A were happening over different seasons. And so to do this, I utilized the modeled data and I averaged the chlorophyll concentrations over an annual cycle for each decade of the time series. And we see that overall, there's been a bimodal pattern in chlorophyll A distribution with one major bloom in the winter time and then one larger bloom in the summertime. But these blooms have been declining over time, particularly the winter spring bloom. We do interestingly see that the last decade, we've seen an uptick in chlorophyll A concentrations. But overall, we see that the blooms are becoming less intense in both seasons over the time series. This then leads me to my next question. Has the timing and duration, or the phenology in phytoplankton blooms, changed over time? And To answer this question, I needed to define phenology metrics for looking at a bloom and defining a bloom. And I scoured the literature to look at different ways to measure phenology. And the best method I identified for this particular time series was to utilize a greater than 5% median threshold, which was established by Siegel and colleagues in 2002. And this method essentially takes the median value of chlorophyll for each year. So for example, I'm showing you the chlorophyll A distribution for 2016. And then whenever the concentration of chlorophyll exceeds that of 5% greater than the median value, it's considered a bloom. So for example, we see that the start of the first bloom in 2016 occurs around week 5 when the chlorophyll concentration first passes this greater than 5% median threshold. And then the end of the bloom is considered to occur around week 9 when that concentration first drops below the 5% median threshold. And then we consider the bloom duration to be the difference between that end date and the start date. And I determined these different bloom metrics for the winter season as well as for the summer season. In terms of results, we saw large variability in the bloom phenology in winter. Just to give you a sense of that variability, we have the bloom start week, the week of the bloom maximum, and then the number of weeks the bloom lasted each year. And we see a lot of variability, but overall no long-term directional change in winter bloom phenology. To summarize these data more, I have box plots here, which again show the start, max, and duration. And we see that the bloom start date occurs typically around week 3, but can range between week 1 and week 12, so all the way from January to the end of March. We see that the week of the bloom maximum occurs around week 5, but can vary between the first week of January to the beginning of April. And we see that the bloom duration is typically around 3 weeks, but can last as little as 1 week and last as long as 2 months. In summer, we saw similar results in that there was no long-term directional change in the timing of the bloom start or the bloom maximum, but we saw that the summer blooms are getting significantly shorter. This figure here shows the timing of the bloom start date in summer, which typically occurs around week 24, but ranges from week 20 to week 27. We see that the bloom maximum typically happens 1 week after the bloom start date at week 25, but ranges between week 23 and week 31. And the bloom duration is getting shorter, such that the first 20 years of the time series from 1968 to 1994, the blooms were around week 6, or lasted around 6 weeks, versus from 1999 to 2019, the blooms are now lasting only 3 weeks. And this is just to show you the actual data and what that looks like over time. And again, we see a lot of variability, but a slight directional change in that the blooms are getting shorter over time. I then wanted to look at the bloom frequency and whether the number of blooms has changed. And what we found was that the number of blooms in summer are significantly greater than the number of blooms in winter, but we didn't find a significant long-term change in the number of blooms over the time series. And so typically we see around 3 blooms in winter, around 4 to 5 blooms in summer, and that overall represents around 7 blooms total in a full year, but the number of blooms can range from 3 in a year to up to 12 in a year. And so overall, these results of no long-term change in phenology or frequency, but strong variability alludes to some of the conclusions that were made by Thibodeau, who started this time series 20 years ago. And the fact that we're still seeing this large variability, I think really speaks to the flexibility and resiliency of the system in some ways. Scott Nixon in 2009 did see some indications of earlier blooms in his time series analysis, but he was using phytoplankton counts, which is slightly less robust than the chlorophyll A biomass data we had here. And we now have 50 years of data to give us a much more holistic picture of what's been going on with the bloom dynamics in Narragansett Bay. This then led me to my third question of what environmental factors might be explaining this variability we're seeing in bloom phenology. And so to do this, I utilized a variable selection method based on a dissimilarity-based method called BioEnv. And so essentially what this method does is it allows me to use a suite of environmental variables and identify what combination of those variables best explains the bloom phenology we're seeing by season. And so this table here shows the different environmental parameters I used. I had climate indices, including the North Atlantic Oscillation and the Gulf Stream Index, as well as nutrients in both winter and summer, temperature and salinity in winter and summer, as well as light and water clarity as determined by Secchi depth. I then looked at the suite of environmental parameters to identify what combination best explained the different phenology metrics in winter. And I found that multiple environmental factors explain the bloom phenology variability we're seeing. Temperature had an important effect on all three bloom phenology metrics in winter. As an example, this is a figure with winter mean temperature on the x-axis and the bloom duration on the y-axis in weeks. And we see that bloom duration is shorter when mean winter temperature is warmer. In summer, I again found that multiple environmental factors affect summer bloom phenology, including climate indices and the timing of different nutrient maximums and salinity. And this figure here shows mean salinity on the x-axis and bloom start date on the y-axis. And we see that typically a higher salinity corresponds to a later bloom start date. And this could have something to do with the timing of the mixed layer depth and when that's enabling phytoplankton to be circulated above their critical depth in bloom, but it's something I still want to look into more in terms of the mechanisms. So overall, we see that we now have 60 years of chlorophyll A and nutrient data for the Narragansett Bay to get a more holistic picture of the long-term changes we're seeing. Some of those changes include a long-term decline in chlorophyll A, both in terms of its magnitude and intensity. We've seen large variability in the bloom phenology over time, but no long-term directional change. And I had the opportunity this summer to work on visualizing this data and these results in a creative way by collaborating with artists from the Rhode Island School of Design. And what I was able to do is utilize the actual annual chlorophyll concentrations that I showed you earlier and print them onto these giant panels of wallpaper and then utilize actual vector microscopic images from the time series and convert them into vector graphics. So then I could create repetitive patterns of these phytoplankton occurring over time. And so what I'm trying to show with this mural is that we're not only seeing a decline in phytoplankton, but we're also seeing changes in the types of the phytoplankton and potentially the distribution of that chlorophyll over time. And this is all superimposed over long-term warming trends that are occurring as we move from blue in the 1970s up to red in the 2010s. And building off of some of these conclusions, and as I mentioned earlier, one question I still have, or one thought, is whether this large variability that we see in these bloom phenology is actually aiding in the stability of the system, in that the plankton are now so flexible in responding to these long-term trends in temperature that it doesn't significantly affect their ability to reproduce and to bloom every year. We saw that phenology correlated with multiple environmental factors, particularly temperature in winter and salinity in summer. And by understanding phytoplankton bloom dynamics, we can have a better understanding of ecosystem-wide changes that might be associated with ocean warming. And so particularly thinking about the bottom of the food web and whether these
this large variability we see in phytoplankton, how that relates to zooplankton biomass, as well as the timing of fish occurrence in Narragansett Bay. In terms of future directions, I would like to apply this question of phenology to phytoplankton community composition and whether there have been significant shifts in different phytoplankton species that might help to explain some of the variability we're seeing in the chlorophyll A data. And so as an example, this figure here is showing the Thalassiocyra percent abundance from December 2008 to December 14, and we see that there's lots of variability in the abundance of this group over less than 10 years. And so I'm hoping to then apply
those data to this chlorophyll A time series and see if it can help us to further elucidate some of these changes we're seeing over time. And with that, I would like to thank the National Science Foundation and the Office of Polar Programs, which funded my research for my PhD, as well as the lab of Debbie Steinberg, who I was mentored by during my PhD. And I'd like to thank the Rhode Island Coastal Ecology Assessment, Innovation, and Modeling Program, which is funded by the National Science Foundation. They're a Rhode Island EPSCoR program, and they're currently funding my postdoc here at URI. And I'd like to thank my current lab in phytoplankton ecology, which is led by Tatiana Rainierson here at the Graduate School of Oceanography. I began my postdoc in January 2020, so have spent most of it at home, but I really appreciated the community that I've established even virtually during this time. And I'd also like to thank a number of people from the Graduate School of Oceanography that helped to maintain this plankton time series I'm analyzing. Ted Sameda, who began it in 1959, and his former student, David Borkman, who's now at the Department of Environmental Management. And he's been really helpful in helping me actually track down some of the old data from the 60s. And the statisticians, Jacob Strzok and Gavino Pugioni, who helped me with the dynamic linear models, as well as all of the plankton assistants that continue to sample the time series every year. And I have my contact information, and I'm happy to take any questions. Thank you very much.
Thanks, Tricia. I have a question, but I will let somebody else go first since I already know some of the answers to questions that might be asked. Yeah, Uta, go ahead.
Yeah, really great talk. Thanks so much. That was really amazing. I want to specifically ask about the theropods that you found in the traps. I don't know if you looked at all, but did you see any indication of damage to their shells from ocean acidification or not?
Yeah, I did have the opportunity to look at some shells that we caught in our nets to see if there was any in situ damage, and it was hard to tell with all of the tissue whether the shells were damaged due to predation or if it was from dissolution. And I decided purposely to not focus on that quite as much since I knew other groups had looked at it at the time. In terms of the sediment trap, I didn't look that closely at those shells either, mainly because I was somewhat concerned about the preservative we use to fix the shells or the animals that fall into the trap, because there has been some evidence that some of the chemicals that are used might further dissolve the shells to a point that it would be hard to tell whether it was a natural process or if it was occurring in the trap itself.
Yeah. What did you use to fix the traps?
It was I believe it's, oh my gosh, I'd have to look back at my methods. I think,
don't worry about
it. Sorry.
Thanks.
I'll go ahead and ask one of the questions that I had. For the drivers of the summer bloom, you had one of the things that was common was the peak phosphate. Do you have any ideas about that?
I know, I'm still trying to think more about some of the inferences or mechanisms that might be at play. I mean, I think one of the things I'm realizing with estuaries is that there's so much variability. It's really hard to parse out some of those, like, really, it's often not a silver bullet in terms of the environmental factors that can explain this variability. Sorry, I'm mumbling a little bit, but I think in terms of nutrients, I mean, phosphate could be a potentially limiting factor for phytoplankton blooms. So maybe we're seeing that the timing of that phosphate peak affects the ability of the phytoplankton themselves to bloom. And so maybe that timing when it's occurring might then affect the timing of when the bloom itself occurs. But this is also a part of estuarine ecology that I feel like I'm still getting a grapple on. So I need to do some more research into those potential mechanisms.
Okay, thanks, Tricia.
Yeah.
Are there other questions?
Thank you.
Uta, do you have more?
I have another one if nobody else.
Yeah.
So your phytoplankton work shows that there was a decrease in chlorophyll overall, which is something which has been found in other places worldwide too. But if you think about the effects of climate change, you can't really predict that necessarily globally it should be a decrease. Do you have a good feeling of what you think globally is happening? Is it a decrease, or are we getting to the point where we can say, yeah, it's decreasing, or not quite yet?
Oh, geez. I mean, I feel like overall, what I've read, and I mean, the Narragansett Bay is interesting because this is a very human-induced estuary. So it's hard for me to parse out how much of some of these declines we're seeing are due to shifts, for example, in nutrient wastewater treatment versus more large-term climate change, like the long-term temperature increases. I mean, I feel like from what I've done, what I've analyzed, it looks like it's probably a bit of both, which seems to always typically be the case with ecology. But at least from what I've read and what I've heard is the idea that with these warmer temperatures, we're going to see increased stratification, which would essentially lead to a decline in phytoplankton blooms. So I would say overall that I feel like I have a tough time being definitive, but I would lean towards thinking less productive oceans overall.
Yeah, I guess it is getting in that direction. I mean, yeah, it's interesting. Thank you.
Mm-hmm.
So I have a question about just the mechanics of the sediment trap collections of pteropods. You indicated it was more of a behavioral thing, right? So that they happen to be sinking and then they see this large yellow thing and they then sort of curl up and die. So already people that work with sediment traps are sort of, they belong to a guild in terms of, what aspect ratio you use and whether or not you tether it and you moor it and different currents and whatever. So you've presumably observed this behavior. How reliable is it to be doing these counts in this way? Do you know?
So that is one of the reasons that I decided to go with this question of phenology more so flux, because there are definitely some inherent biases with the different types of sediment traps you use. And actually one project I worked on during my PhD, but it never was finished while I was there, was we had two sediment traps deployed, one that was a cone and one that was a column. And we were looking at the differences in the capture of zooplankton and also just particulate organic carbon flux. And at least my perception from counting and quantifying the samples was that you definitely tended to get more flux in the cone-shaped trap. But again, we haven't statistically analyzed it, so I'm not entirely sure if that's the case. But in terms of this pteropods that I was measuring, We think that they were alive because there was tissue in almost all of the pteropods that we collected, versus sometimes, occasionally, we would get shells that didn't have any tissue in them. So that would indicate to me that it had been eaten by a shellless pteropod, which they eat the shelled ones, and then the shell would sink into the trap. And zooplankton swimmers are a pretty common occurrence in these sediment traps. But again, that was one of the reasons that I wanted to look more at this question of phenology, because then I could just measure like a subset of 100 per cup and not think more about and not focus as much on the absolute values of the fluxes over time.
Well, good for you for being so careful. I think many people would have actually said, okay, well, I've got a time, I've got an area, let's call it a flux. So good for you for your honesty there. But I'm sure that at some point those flux data could be used once you can figure out some of the controlling factors behind it, perhaps.
Yes, and there is actually a colleague from Germany who has been working on the flux data, and it is useful, especially when we want to try to look at particulate and organic carbon in relation to particulate organic carbon. So that work is definitely being done, but yeah, I focused more on, I thought the phenology question was more interesting anyway, so that was sort of the way that I went.
Yeah, it was a great story. Thanks.
Thank you.
Okay, are there any more questions? Because if not, we can go ahead and end the seminar. Thank you, Tricia. I really enjoyed the talk.
Yeah, thank you for the opportunity, Colleen. I very much appreciate it.
Yeah, definitely. Okay, well, thank you everybody for coming. And again, thanks to Tricia for speaking to us this week. Nice to have you all here.
Great.
Yes, thank you, Tricia.
Thank you.
Yes, this was really great.
Great, thank you. Great to see you.
Enjoy your new home.
Yes, thank you.
Bye.
Bye.
Bye.